So in our last video, we talked about how cellular respiration occurs, and it's how the cell is going to bring in something, sugar, and it's going to break it down through this process and start to harvest energy from it. The slower and more steps we can do this in, the more we can harvest from it. If we did it only in one big fast step, we'd be able to harvest only a small amount of energy from that and reaction, and we'd lose a lot as heat. So by doing it in multiple steps really slowly, we can harvest a ton of energy from it, and we can make more ATP. That's why there's so many different steps involved here. And most of them are enzymes working in the background doing this work. So this first step is called glycolysis. It's called lysis, means breaking something down, and gly means sweet, glycus. And this is going to be a step that really doesn't need energy, need oxygen. Everything else is going to require oxygen to work, but this is the only step that doesn't require oxygen. It occurs in the cytoplasm, which is that liquidy part of the cell. So if we just go past the lipid bilayers, there's going to be all of the different organelles, and the organelles swim in the cytoplasm, the liquidy part. It's basically called substrate level phosphorylation, where we're going to take enzymes and sort of harvest phosphorus and sort of put it onto another molecule, and we'll talk about how that works in a second. And there's two main parts. There's going to be the ability to break glucose down and then extract some energy from it. So here's the big picture. Glucose comes into the cell, and it's 1, 2, 3, 4, 5, 6 carbons. So let's go ahead and write that down. 1, 2, 4, 5, 6. And then what happens is there's going to be an enzymatic reaction that splits this in half. And that splitting of that glucose molecule in half basically goes ahead and makes two molecules that are called pyruvate. And these pyruvate molecules basically during the splitting process has released a little bit of energy. Anytime we break some of these carbons off, we're able to get some energy from it. And what it does is it takes this thing called NAD, which is kind of like what I call a wheelbarrow. And a wheelbarrow, you can add some stuff to it and take it somewhere else, and it dumps that payload somewhere. So NAD is going to pick up 2 electrons and hydrogens, and it's going to take it somewhere else. We'll talk about that one in our next couple of videos. So basically what we've done is we've turned NAD into NADH. The other thing we're able to do is we're taking this lower energy form called ADP, and the D is the most important part. It's the diphosphate. So this stands for adenine diphosphate, which means just 2 phosphates. And this reaction is able to go ahead and take an extra phosphate and stick it here. So that becomes the high energy form ATP, and the T just stands for triphosphates. So here we have 2 phosphates. This is the low energy form. Here we have 3 phosphates. And this is the high energy form. It could go somewhere now in the cell, and how it does work is it breaks off this phosphate, and a little bit of energy is released. That energy allows the cell to do some work, and then what happens is this comes back as the diphosphate plus 1 loose phosphate, and this step or a couple other steps will basically glue them together to come back to the ATP form. In order for glycolysis to start, you must put energy in, and this process is just like that. We need energy in to actually make a bunch of energy. And you probably heard that term, it takes money to make money. Well, it takes ATP to make ATP. Specifically, we need 2 ATPs to get this process started. So what we're going to do is, if you look at my next slide, you'll see that we have our 6 carbon molecule. It costs us 2 ATP to break it in half. And then what happens is, through this process, we've harvested some energy. Those phosphates that did get put on there to break it half, we can harvest those. Plus the splitting of this molecule makes it so we can actually make 4 ATP energized. And our new sugar is going to be called pyruvate. We still record this as only 2 ATP made because we needed 2 ATP to make 4 ATP. So this would be kind of like the gross, kind of like with your paycheck, you get a gross amount. And then 2 would be the net. And we only really record it as the net because if it costs you 2 to make 4, we just say 2. So for instance, if you had a job and you made $20 today, but it cost you $10 in gas to get there, you don't say you made $20. You have to account for that gas money. So you say you actually made $10 today. And that's what we're doing here. The other thing it does is it makes that NAD become NADH. And NADH is basically just going to gather up some hydrogens and electrons and move it to another step later. So our net gain is 2 ATP. Our next step is called the preparatory phase. And basically what happens in this phase is we have to take that pyruvate and manipulate it just a little bit to get it into the mitochondria. So we're still outside of the mitochondria. This one requires air, so it's aerobic. It starts out in the cytoplasm and ends up in the mitochondria. And the whole point is to basically get some more carbons into the Krebs cycle. So what happens is we have that pyruvate. Remember how we had actually another pyruvate because that glucose got split into 2 of these? But let's only just do the fate of 1 pyruvate just to make our lives a little easier. So here's our 1, 2, 3 carbon molecule. What happens is we have a bunch of enzymes that is going to peel off one of these carbons. And that way what happens is we still have a 2 carbon molecule come out here. And it gets attached to this enzyme. And this enzyme is called acetyl-CoA. And basically what this guy does is it's able to deliver this 2 carbon molecule into the mitochondria. Without this enzyme, these 2 would just float around in the cytoplasm and do nothing. So basically what has to happen is we have to convert it into a 2 carbon molecule, attach it to acetyl-CoA, and acetyl coenzyme A will bring it into the mitochondria, and we're going to be able to do some work. But in the meantime, we've done some things. One of these carbons broke off, and we added oxygen to it. That's why it's an aerobic step, and it became CO2, gas. And that gas is going to go to our lungs. Then what's going to happen in this step is because every time we break off a little carbon, we're able to make some energy. And this energy is not going to be ATP. It's going to be just to take that empty wheelbarrow, another NAD, and add 2 electrons and some hydrogen to it. And now what's going to happen is they're going to go to our next couple of steps that we'll talk about. So really the only thing that we've done here is 1, we've arranged pyruvate to become a 2 carbon sugar and attached it to an enzyme so it can go in the mitochondria. We were able to release one of the CO2s into the bloodstream and eventually it'll go to the lungs. And we've taken one of those empty wheelbarrows, NAD, and chucked 2 electrons and some hydrogens in it, and it's on its way to a step that we'll talk about soon. Our inputs for this one is going to be 2 pyruvates, which is 3 carbons, 2 NADs, the empty wheelbarrows, and 2 coenzyme A's, which are basically going to drag them into the mitochondria. Our outputs are going to be 2 acetyl-CoAs attached to the 2 carbons, 2 NADHs, and then 2 CO2 molecules that will just leave and go out to our lungs. So pyruvate comes in, just a quick recap. One of the carbons leaves as CO2. We have this 2 carbon molecule now. it gets attached to acetyl-CoA and moves into the mitochondria. Through this process, we're able to energize two NADHs. So we had an NAD, we added some hydrogens and electrons to it, became NADH. And the two carbons go to the Krebs cycle, which is our next cycle. And the other ones just kind of get expelled out as waste. So far this is what we've done. We've taken glucose, we've turned it into pyruvate. This happened outside the cell, outside the mitochondria, excuse me. So this is in the cytoplasm. And finally, we're inside the mitochondria, and we're going to go to our next step. It's called the citric acid cycle, Krebs cycle, TSA cycle, and this is where we're going to make some more energy. I'll talk about that in my next video.